Welcome. Today we're diving into CAR T-cell therapy, a groundbreaking approach in cancer treatment that's changing the landscape for patients with difficult-to-treat diseases. This therapy harnesses the power of a patient's own immune system, specifically T cells, to target and destroy cancer cells. Over the next several scenes, we'll explore how CAR T-cell therapy works, its manufacturing process, approved uses, potential side effects, and what the future holds for this innovative treatment. Let's start by understanding what makes CAR T-cell therapy so unique and why it's considered a major advancement in modern medicine. CAR T-cell therapy stands for chimeric antigen receptor T-cell therapy. In simple terms, it's a type of adoptive cell therapy where a patient's own T cells are collected, genetically modified in the lab to recognize cancer cells, and then returned to the patient's body. The engineered T cells are now equipped with a synthetic receptor, called a CAR, that allows them to seek out and destroy cancer cells more effectively. This process bypasses some of the limitations of the natural immune response, making it a powerful tool against certain cancers. Chimeric, part of CAR T-cell therapy, refers to the unique structure of the receptor added to the T cells. This receptor is built by fusing different protein components, an antigen recognition domain from an antibody, a transmembrane domain, and one or more intracellular activation domains from T cells. This fusion allows the engineered T cells to recognize specific markers on cancer cells and become activated, regardless of the usual pathways that control T cell activity. This innovation is what gives CAR T cells their remarkable targeting ability. First step in CAR T-cell therapy is leukapheresis, a process where a patient's blood is drawn and passed through a machine that separates out the white blood cells, including T lymphocytes. The rest of the blood is returned to the patient. This outpatient procedure usually takes 3 to 4 hours and is generally well tolerated. Collecting these cells is essential, as they will be the foundation for the personalized CAR T-cell product that will later be infused back into the patient. Once the T cells are collected, they're taken to a specialized laboratory for genetic engineering. Here, scientists use viral vectors, often disabled retroviruses or lentiviruses, to insert the CAR gene into the T cell's DNA. This process ensures that the T cells will express the synthetic receptor needed to recognize cancer cells. Non-viral methods, such as transposon systems or CRISPR-based knock-in techniques, are also being developed to improve efficiency and safety. This step is crucial for giving the T cells their new cancer-fighting abilities. After genetic modification, the engineered T cells are placed in culture and expanded outside the body. This process typically takes 1 to 2 weeks and involves stimulating the cells with cytokines like IL-2, IL-7, or IL-15. The goal is to grow enough CAR T cells, often hundreds of millions or even billions, to ensure a potent therapeutic effect. This expansion phase is carefully monitored to maintain cell quality and function, setting the stage for the next steps in the treatment process. Before the engineered T cells are infused, patients typically receive a short course of lymphodepleting chemotherapy. This regimen, often using drugs like fludarabine and cyclophosphamide, reduces the number of existing lymphocytes in the body. By doing so, it creates space for the new CAR T cells to expand and function more effectively. This step also helps reduce immune rejection and competition for cytokines, improving the chances of successful engraftment and anti-tumor activity. Preparation complete, the patient receives an infusion of their personalized CAR T-cell product, usually as a single dose. Once inside the body, these cells engraft and begin to expand over the next 1 to 2 weeks. They travel to sites of disease and start attacking cancer cells. In some cases, CAR T-cells can persist for months or even years, providing ongoing surveillance against cancer recurrence. This phase is closely monitored to ensure safety and effectiveness. Once the CAR T-cells encounter their target antigen on a tumor cell, they become activated and form a specialized connection called an immunologic synapse. The CAR T cells then release cytotoxic granules containing perforin, which creates holes in the tumor cell membrane, and granzymes, which trigger cell death. Additionally, CAR T cells secrete pro-inflammatory cytokines that help clear tumors, but can also contribute to side effects. Remarkably, a single CAR T cell can kill multiple tumor cells in succession. As CAR T cells attack tumor cells, they release a surge of cytokines, signaling molecules like IFN gamma and TNF alpha. These cytokines help recruit other immune cells and amplify the anti-tumor response. However, high levels can also cause systemic inflammation, leading to side effects such as fever and low blood pressure. The ability of CAR T cells to engage and destroy multiple tumor cells, known as serial killing, is a key advantage of this therapy. Currently, 7 CAR T cell products have received FDA approval. These therapies target either CD19, a marker found on B-cell malignancies, or BCMA, which is present on multiple myeloma cells. Each product is approved for specific cancer types, including pediatric and adult B-cell acute lymphoblastic leukemia, large B-cell lymphoma, mantle cell lymphoma, chronic lymphocytic leukemia, and multiple myeloma. The list of approved indications continues to grow as research advances. Zigenlecleucel, marketed as Kymriah, and axicabtagene ciloleucel, known as Yescarta, are 2 of the most widely used CAR T-cell therapies. Kymriah targets CD19 and is approved for pediatric and young adult relapsed or refractory B-ALL, as well as adult large B-cell lymphoma. Yescarta also targets CD19 and is approved for large B-cell lymphoma, follicular lymphoma, and other indications. These therapies have paved the way for broader use of CAR T-cell treatment. Exucabtagene autoleucel, or Tecartus, and lisacabtagene maraleucel, known as Breyanzi, are additional CAR T-cell therapies targeting CD19. Tecartus is approved for mantle cell lymphoma and adult B-cell precursor ALL, while Breyanzi is used for large B-cell lymphoma and chronic lymphocytic leukemia. Each product is tailored to specific patient populations, reflecting the precision of CAR T-cell therapy. Ecubtagine viclucil, sold as Abecma, and ciltacabtagene autoleucel, known as Carvykti, are CAR T-cell therapies that target BCMA, a marker found on multiple myeloma cells. Abecma is approved for patients with relapsed or refractory multiple myeloma who have received at least 2 prior lines of therapy. Carvykti is indicated for those with at least 1 prior line, especially if they are refractory to lenalidomide. These therapies offer hope to patients with limited options. Indications for CAR T-cell therapy are expanding rapidly. For example, lisacabtagene maraleucel and other CD19-targeted products are now approved for relapsed or refractory follicular lymphoma after at least 2 prior lines of therapy. As clinical trials continue, more cancer types and patient populations are expected to benefit from CAR T-cell approaches. It's important to consult the latest FDA prescribing information for the most up-to-date indication. One of the most common and important side effects of CAR T-cell therapy is cytokine release syndrome, or CRS. This occurs when the infused CAR T-cells become highly activated and release large amounts of cytokines such as IL-6 and interferon gamma. Symptoms usually appear within the first 1 to 2 weeks after infusion and can include fever, low blood pressure, and low oxygen levels. CRS is graded on a scale from 1 to 4, with higher grades indicating more severe symptoms. Managing cytokine release syndrome depends on its severity. Mild cases can often be handled with supportive care, such as fluids and fever reducers. For moderate to severe cases, tocilizumab, an IL-6 receptor antagonist, is commonly used to block the effects of cytokines. If CRS does not improve, corticosteroids may be added. Early recognition and prompt treatment are essential to prevent complications and ensure patient safety. Another key toxicity is immune effector cell-associated neurotoxicity syndrome, or ICANS. This condition can occur alongside or after CRS and affects the nervous system. Symptoms may include confusion, difficulty finding words, tremors, reduced attention, and in severe cases, seizures or brain swelling. Monitoring for ICANS involves regular neurological assessments, such as the ICE score, during the high-risk period after infusion. Mainstay of ICANS management is corticosteroids, which help reduce inflammation in the brain. Anti-seizure medications may also be used as a preventive measure. Unlike CRS, tocilizumab is less effective for ICANS because it does not penetrate the central nervous system well. Careful monitoring and early intervention are critical to minimizing the risk of serious complications. Beyond CRS and ICANS, patients may experience other side effects. Prolonged cytopenias such as low neutrophil or platelet counts are common and can last for weeks or months. CD19-targeted therapies may cause B-cell aplasia and low antibody levels, sometimes requiring intravenous immunoglobulin replacement. Infection risk is increased due to immune suppression, and rare cases of secondary malignancies have been reported, leading to long-term monitoring requirement. To the risks associated with CAR T-cell therapy, treatment is only provided at certified centers. These facilities have specialized REMS programs, 24/7 monitoring, and immediate access to medications like tocilizumab and intensive care support. This ensures that any complications can be managed quickly and effectively, providing the safest environment for patients undergoing this complex therapy. CAR T-cell therapy has shown remarkable success in patients with relapsed or refractory cancers who have exhausted standard treatments. Clinical trials like ZUMA-1, Juliet, and Transcend for lymphoma, and CAR MMA and CR8UD for myeloma have demonstrated high rates of complete remission. Many patients experience durable responses, with some remaining cancer-free for years. This durability is a key advantage over previous therapy. What truly sets CAR T-cell therapy apart is its potential for long-term remission or even cure in a subset of heavily pretreated patients. Unlike traditional salvage therapies, CAR T-cell treatments can provide lasting immune surveillance, reducing the risk of cancer returning. This has led to CAR T-cell therapy being used earlier in the treatment course for certain high-risk cancers, based on evidence from trials like ZUMA-7 and Transform. CAR T-cell therapy isn't just for cancer anymore. Researchers are now exploring its use in autoimmune diseases like systemic lupus erythematosus and antisynthetase syndrome. Early studies with CD19-targeted CAR T-cells have shown promising results, including drug-free remission in some patients. This could open new doors for people with severe treatment-resistant autoimmune conditions. One of the most exciting developments is the creation of allogeneic or off-the-shelf CAR T-cell products. These therapies use donor-derived, gene-edited T-cells, which can be manufactured in advance and stored for use when needed. This approach could dramatically reduce the time from patient evaluation to treatment, making CAR T-cell therapy more accessible and efficient for a wider range of patients. While CAR T-cell therapy has revolutionized blood cancer treatment, its use in solid tumors remains a major challenge. Solid tumors often have diverse antigens, a suppressive microenvironment, and barriers to T-cell entry. Researchers are working to overcome these obstacles by developing new CAR designs and combination therapies. Success in this area could expand the reach of CAR T-cell therapy to many more cancer types. Next-generation CAR T-cell therapies are in development, featuring advanced designs like dual antigen targeting and armored CARs that co-express cytokines. Some approaches aim to engineer T-cells directly inside the body, known as in vivo CAR T generation. These innovations could enhance effectiveness, reduce side effects, and broaden the range of treatable diseases. Despite its promise, CAR T-cell therapy remains out of reach for many eligible patients. The manufacturing process is complex and centralized, requiring travel to certified centers. The cost per infusion can be several $100,000, not including hospital stays and management of side effects. These barriers highlight the need for expanded manufacturing capacity and more efficient delivery model. Efforts are underway to make CAR T-cell therapy more accessible. Strategies include increasing manufacturing capacity, shortening the time from cell collection to infusion, and developing allogeneic products that don't require patient-specific cells. These changes could help more patients benefit from this life-changing therapy, regardless of where they live or their financial situation. Clinical trials have been essential in advancing CAR T-cell therapy. Studies like ZUMA-1, Juliet, Transcend, and others have provided the data needed for FDA approvals and expanded indications. Ongoing trials continue to refine dosing, improve safety, and explore new disease targets. Participation in clinical trials remains a key way for patients to access cutting-edge treatments. Not every patient is a candidate for CAR T-cell therapy. Careful screening is required to determine eligibility, including assessment of disease type, prior treatments, organ function, and overall health. Disparities in referral and screening can limit access, making it important for healthcare providers to stay informed about evolving guidelines and ensure equitable care. Journey through CAR T-cell therapy is complex and requires coordination among multiple specialists. From initial consultation and cell collection to infusion and post-treatment monitoring, each step is carefully managed. Patients and families receive education and support throughout the process, helping them navigate the challenges and maximize the benefits of this advanced therapy. After CAR T-cell infusion, patients are closely monitored for several weeks. This includes regular blood tests, imaging studies, and neurological assessments to detect early signs of toxicity or relapse. Most side effects occur within the first month, but long-term follow-up is essential to monitor for late complications and ensure sustained remission. Long-term outcomes with CAR T-cell therapy are encouraging, especially for patients with few other options. Many achieve complete remission and some remain disease-free for years. However, ongoing research is needed to better understand who benefits most, how to manage late toxicities, and how to further improve outcomes for all patients. Hearing from patients who have undergone CAR T-cell therapy can be inspiring. Many describe the process as challenging but ultimately life-changing, especially for those who had exhausted all other treatment options. Their experiences highlight the hope and potential that CAR T-cell therapy brings to the field of cancer care. Successful CAR T-cell therapy program relies on a dedicated healthcare team, including oncologists, nurses, pharmacists, and support staff. Each member plays a vital role in patient education, monitoring, and management of side effects. Teamwork ensures that patients receive the safest and most effective care possible throughout their CAR T-cell journey. Future of CAR T-cell therapy is bright, with ongoing research focused on improving safety, expanding indications, and making the treatment more accessible. Innovations like allogeneic products, next-generation CAR designs, and new disease targets are on the horizon. As our understanding grows, CAR T-cell therapy is poised to benefit even more patients in the years ahead. Education and advocacy are essential for patients considering CAR T-cell therapy. Understanding the risks, benefits, and logistics of treatment empowers patients to make informed decisions. Advocacy groups and educational resources can provide valuable support, helping patients and families navigate the complexities of advanced cancer care. Navigating insurance coverage and financial support is a significant part of the CAR T-cell therapy journey. Many centers have dedicated staff to help patients understand their benefits, apply for assistance programs, and manage out-of-pocket costs. Addressing financial barriers is crucial to ensuring that all eligible patients can access this life-saving treatment. CAR T-cell therapy is subject to rigorous regulatory oversight to ensure safety and efficacy. The FDA reviews clinical trial data, approves new products, and monitors for long-term risks such as secondary malignancies. Certified treatment centers must follow strict protocols and participate in REMS programs to maintain high standards of care. Long-term follow-up is essential for patients who receive CAR T-cell therapy. This includes regular checkups to monitor for late side effects, infections, and potential secondary cancers. Ongoing surveillance helps ensure that any issues are detected early and managed promptly, supporting the best possible outcomes for survivors. Progress in CAR T-cell therapy is the result of collaboration among scientists, clinicians, industry partners, and patient advocates. Ongoing innovation is driven by shared knowledge and a commitment to improving patient outcomes. As new discoveries are made, the field continues to evolve, offering hope to more people facing serious diseases. Recap: CAR T-cell therapy is a personalized, highly effective treatment for certain cancers and possibly autoimmune diseases. It involves collecting and engineering a patient's T-cells, expanding them and reinfusing them to target disease. While the therapy offers hope for long-term remission, it comes with risks and challenges, including side effects, cost, and access barriers. Ongoing research and innovation are making this therapy safer and more widely available. As we look to the future, CAR T-cell therapy is set to play an even bigger role in cancer and immune disease treatment. With advances in manufacturing, new targets, and improved safety, more patients will have access to this life-changing therapy. The journey is ongoing, but the progress so far is a testament to the power of science and collaboration. Thank you for joining this comprehensive overview of CAR T-cell therapy. From its innovative mechanism to its expanding uses and ongoing challenges, CAR T-cell therapy represents a new frontier in medicine. As research continues and access improves, this therapy will bring hope to even more patients and families. Stay informed, and let's look forward to the next breakthroughs in this exciting field.